Alright, ladies and gentlemen, this is going to be the next video on biology, and specifically going over the overview of the central dogma. We want to go through and understand what the central dogma is, and how it goes through and applies to proteins. Let's get started on this next video. So what is the central dogma, and what does that terminology and that particular vocabulary word actually go through and mean? Well, the central dogma is the molecular process of going from DNA to RNA to protein. And the term was actually coined by Francis Crick, somebody we've already gone through and looked at already. Now, in his own words, he called this the idea of the central dogma for two reasons. He had already used the obvious word hypothesis in the sequence hypothesis. In addition, he wanted to suggest that the new assumption was more central and more powerful. So let's go through and look at the overview of the central dogma here. What we're going to have initially here is we have our DNA that we're going to go through. And we're going to go through and try to use an enzyme called RNA polymerase. We're going to go through and take our DNA, unwrap a section, and we're going to make RNA from that section. This process here, the very beginning, is called transcription. And this process here is what we're going through when we take DNA and convert it into RNA. Now, when we go from RNA into our proteins, we're going to be going through and we're going to call this translation. Translation is also going to be used via enzymes and ribosomes and all sorts of other things that we'll learn about. We want to break this entire process down into two chunks. We want to break it down into transcription and translation. All right, so what is transcription then? If we're looking at the definition for transcription, well, transcription is the first part of the central dogma, and it's going to be going from DNA to RNA. So we want to go through and take our DNA out of our nucleus, create a stable, in this case, messenger RNA molecule. We're going to use that messenger RNA molecule later to go through and get the desired protein that we need. But we're taking that coded information and going from DNA to RNA. All right, so if we're going through and looking at this now, well, what is translation? Well, translation is going to be the second part of the central dogma. It's going from that RNA into that protein. So we're going to take our RNA, our mRNA, and our tRNA, which we'll learn about in a little bit. And we're going to go through and assemble our amino acids in that polypeptide chain. And that polypeptide chain is going to make our desired protein. So translation is going from RNA into that protein. Now, there are going to be some key important players when going through and talking about transcription and translation. So let's go through and just look at transcription here. Obviously, we have our DNA and our RNA, but we also have an important enzyme. We have RNA polymerase, which is going to go through and allow this reaction to occur. Our end goal or our product of transcription is going to be this mRNA that we're then going to use in translation. Now, translation has a few more individuals that are going to go through and allow this to occur because the process is more complicated. We obviously have our mRNA, but now we're going to have our ribosomes along with our tRNA and then our amino acids that we put together in our polypeptide chain. Remember, the end goal of transcription is going to be to go through and create that protein and that protein molecule. So if we're looking at the end goal is to make an amino acid sequence, which is formed into a complex protein. We can either do this at ribosomes free floating in the cytoplasm or on the rough endoplasmic reticulum. All right, so did you learn? Well, did you learn a couple of things? Did you learn about what this central dogma is? Did you learn about what transcription and translation are? And then lastly here, did we learn about the end goal of the central dogma? What is the point of us going through and doing this? This will be the end of the video. I will see you all in class tomorrow.